Well, hello and welcome to the latest installment in Datacolor series of microscopy webinars. Morphometry and density intensity readings, avoid errors in quantification. I'm Mark McNulty, General Manager of Datacolor. With me today from Datacolor is Mark Clymer, ChromaCal's Marketing Director. We thank you all for joining us today. For today's webinar, we are also pleased to have Dr. Michael Linden and Jerry Sedgewick, both of whom we'll introduce shortly. Just briefly regarding our agenda, after Jerry's discussion, we'll review some upcoming events and offers from Datacolor, and then open up a question and answer session. But if you have any questions during the seminar, please enter them into the question area of the webinar control panel. We'll either address them today or we'll follow up with you separately. So it's now my honor to introduce Michael Linden. Dr. Linden completed his medical scientist training program at the University of Minnesota, receiving both a PhD and MD. His residency involved anatomic and clinical pathology and hematopathology at the University of Washington. He is currently assistant professor and associate director for hematopathology and laboratory medicine at the University of Minnesota. Dr. Linden's particular clinical and research interests are in flow cytometry and plasma cell myeloma. And in his chosen field, Dr. Linden is a guest lecturer, serves on several editorial boards, and has co-authored book chapters and various peer-reviewed publications. So without further ado, Dr. Linden.
Thank you. It's a pleasure to be part of this. I first got interested in ChromaCal because of Jerry Sedgewick. Jerry and I had previously worked together when I was a PhD student, and he was working here at the University of Minnesota. When I was reacquainted with Jerry at a professional meeting, I got intrigued by this system. So one of the things I do as a pathologist is I do a lot of bright field microscopy, and a lot of the times if I'm publishing something or if I'm taking pictures to share with friends, I'm doing a lot of digital photography. Now, digital photography has made it easier for the novice to collect pictures because I'm not an expert user of cameras like Jerry. But one of the problems that I've noticed is that there's a lot of image inconsistency. So Jerry has this system called ChromaCal that he introduced me to, which is a system that kind of post-process those images and makes them very consistent. So Jerry and I, in conjunction with Dr. Marna Erickson, who's a faculty member here in the Department of Dermatology, did a study where we took some really high-quality stained slides from bone marrow. They included right chemosynth stained slides, immunoperoxidase stained slides, and H&E stained slides. And we basically showed that if you take a bunch of pictures, even if you have an expert person like Jerry getting all the pictures, there's some inherent problems with the camera systems that give a little bit of image-to-image inconsistency. And what we show is that using ChromaCal, it's maybe an easier and an optimal system to post-process those images to make them very consistent in terms of color, tonal elements, etc., than kind of other more commonly used post-processing methods. So next, I'd really like to formally introduce Jerry. So Jerry is a recognized authority on Photoshop and using Photoshop for scientific images, and he's authored 2 books and several other book chapters on this topic. And he's been very well published in this area, has been an author or co-author of numerous articles in peer-reviewed journals, including Science, Circulation, PNAS, etc. And he's directed a core microscopy facility here at the university for more than 15 years. He's taught nationally at the Marine Biology Laboratory in Woods Hole, Massachusetts, and has been part of numerous workshops and presentations on scientific imaging and image integrity at scientific conferences. And now he's actively involved in consulting services on scientific imaging and image analysis through his own company, which is called Imaging and Analysis. So without much further ado, I'd like to present to you Jerry Sedgewick, who will present his very elegant study that he did in conjunction with myself and Dr. Erickson.
Hello and welcome. Let me get my screen looking appropriate here. I really appreciate it that everyone could show up today. I wish I could see you, but I'll do the best I can without being able to see your faces. So today we're going to talk about, and we're going to go through an overview of segmenting, separating regions of interest from surrounding areas for those of you who may not know what that term means. There are two methods that are often used. One, you threshold in order to separate your objects of interest from surrounding areas, from background. That's done by using a tonal value above which you might have tones that would include your objects of interest, and below which you would exclude your surrounding area. You can also do that by perhaps choosing the color of the stain and using color as a method for separating out your regions of interest. We also want to go over some measurement considerations, mostly those that have to do with maintaining linearity, and I'll talk more about that later. We're going to talk about a solution for consistent segmenting with that also includes linear methods in post-processing that Mike's already talked about, called ChromaCal. And you'll see that I've done an experiment to demonstrate the accuracy of measurements after using the ChromaCal method. So for those of you who may not be familiar with this, I'm sure many of you are, when you do the sort of segmenting in which you would separate out your regions of interest from background, generally you start in this instance with a color brightfield image. You create some sort of a black and white image where your stain is darker or lighter than surrounding area, and then you use a sort of threshold to then identify the areas you wish to measure. And these are black against a white background in an image that's called a binary image. You can do the same thing with color. Use color, in this instance brown, for an immunoperoxidase stain, and then from that brown image you can set a threshold. But in the real world, what you find out is that when you take these images, there's a fair amount of variability when from microscope session to microscope session, and sometimes within a single session. And in this instance, this is kind of an egregious example, but in this instance you can see that there's a fair amount of variability taking images. The person who took this image was unfamiliar with the camera, and the major problems have to do with white balancing issues. The background here should be a neutral white, and with varied exposures. And as you can imagine, this is just one user on one camera microscope, and this is just one session using manual camera settings, but imagine what it would be like if you had more than one user, more than one microscope, and more than one session. So now when you segment, things start to become a little more difficult. Now you may have set the threshold to one setting for the perfect ideal image, but once you get an image that's not white balanced correctly, or one that's underexposed, you now have to set a different threshold value in order to include the exact same regions of interest. The same can be said for color segmenting. In your ideal image, you may set the threshold at one level, then you try to match to get the same regions of interest included, and that threshold then becomes a different number. And when they're underexposed, again, the threshold is another setting. To solve the problem of having to set various thresholds and to use a single threshold, you could use methods to make the images consistent, and you could use various post-processing programs, and if you really know how to use them, you can have varying levels of success. But you have to remember that when you do this, it's also very time-consuming. You can create some sort of learning software if you have enough computer programmers at your disposal, and then the software learns what's supposed to be the object of interest. Otherwise, you're kind of left making adjustments to threshold levels manually, or maybe you can reattempt acquisition, but often it's a lot of work to do that, and it's actually, to my way of thinking, and in my own experience, somewhat rare to be able to get perfectly consistent images. during this process, you have to keep in mind that if you're going to do some kind of optical density measurement, some kind of densitometry, that all of your post-processing changes must include a method where you keep the linearity. So when you have differences in exposure, for an example, you're going to have to do some kind of histogram adjustment, and you have to do that correctly. A little bit about linearity. When you take an image, the idea is that you have equal increments of tone as you work your way from dark to light, represented by this graph underneath this particular gradient. If you change that and make your image nonlinear, you no longer have equal increments from dark to light, and you've actually created a scenario in which, were you to measure from this sort of an image, you would actually get erroneous measurements unless you calibrate against a standard. There's also a qualitative side to that. When you're looking at the monitor, if you have or if you're looking at this image in publication, for an example, you would see that there's quite a difference in the way in which you can perceive the dark to the light in the linear image, and actually it's quite another image when you see it and it's nonlinear. And I find it, as an aside, I find it rather surprising that in so many labs you're making these evaluations day after day after day, and so many labs don't have monitors that are calibrated. Having come from a photography background, thinking to myself that in the sciences everyone's going to use things that are calibrated. For an example, you do calibrate a pipette, that in fact in every lab I'd find monitors that were calibrated, but in fact that's the exception rather than the rule. So the requirement then, if you're going to do some kind of optical intensity or density measurement, is that your camera systems need to produce some sort of a linear image, and also in addition to that, images would need to be calibrated against some sort of a standard. And surprisingly, in my wanderings around from lab to lab, I've found with my colleagues at Data Color that we're finding about 50% of color bright field systems that are not producing linear images. These color systems simply don't do that, and it's a tremendous concern to me that some data is getting published that's actually erroneous. So how can you create consistent images and assure linearity? Here's a method that that I'm using now for going about this process, and it's a product that's been out now for about 9 months, and this product includes a calibration slide and then software, and that calibration slide has a matrix of colors against which images are then standardized. This image would be taken at each microscopy session, and then from this image you would then standardize these colors and then apply that standard to images in the software. You can also get this with a monitor calibration, hint, hint. So here is a series of images then. These are immunoperoxidase stained bone marrow, and in this series of images you can see that there are some differences in some inconsistencies in the images that were taken. The first image we'll call the day 1 image. The image that follow vary from that image either by exposure or by the color balance. So if I had this set of images and I wanted to correct these and make these consistent, then I can go into ChromaCal to do that. And what I would do first is open the calibration image. And then once I have that calibration image, I would align it correctly, and then enlarge it and move the grid over the color patches. Now you'll note down in the lower left that there's sort of a quality control set of prompts. And in this instance, it's telling us that our matrix alignment's good, our exposure is good, but most importantly, it's telling us our linearity is good. In other words, it checks to make sure that your camera system is producing linear images. So rather than work with images one by one, I'd rather do things quickly, and so I'm going to batch process then a number of images, and I'm going to batch process those images in really just one step. So here you can see that you have the underexposed image on the left and then the calibrated image on the right, and you can see that it's done that for every image that's in this particular series. So I'm going to go ahead and process those images, and rather than wait for this to complete, which doesn't really take very long, back to the slide presentation, and here is the images that resulted from that process. So you can see that the first image here has been corrected, second image, and so on have been processed in ChromaCal, and that these are very consistent. But I don't know if I believe that, so let's do an experiment. So here are the consistent images that are from ChromaCal. These were segmented in Photoshop, and the threshold was set to a single value for all of these. And to test that against another method for going about getting measurements, in this instance for getting area measurements and a morphometric measurement for area, surface area, what I decided to do was to take the inconsistent images and then set the threshold manually, and then that's a visual affair. And you can see that there's a little bit of a difference here. If you look at this feature here, it's kind of closed up over here. And to try to keep myself from being biased in doing this, I actually would set the threshold level by looking for noise in the background. When that noise was virtually gone, I left the threshold level at that point. And then I did a third method, which would be to take these inconsistent images, apply one threshold setting for all of those so that I can create a sort of control condition. So theoretically, what should happen then is from these images, because it's of the same field, I should get consistent measurements. And here's the results. So you can see that along my y-axis I have the area, and this is in pixels squared, and then each one of these would be the image number. And the magenta is showing my results when I set the threshold manually, and you can see there's quite a bit of variability. And the yellow shows where I've applied one threshold to the inconsistent images, and again you see the variability. But when I apply one threshold to the images with the consistent ChromaCal images, you can see that the variability is not so great. So let's do the same thing doing density measurements. Here are the same set of images. These images, again, were corrected in ChromaCal as before. Only this time, and then from those consistent images, I created a binary image and then inverted it so that I can create a mask, and in that manner I only measure what my features of interest here. And rather than set the thresholds image by image, I instead decided I'm going to test this against a sort of standard in which I did histogram matching. So I took values from this first image and then matched the rest of those images to the first image, and you can see these are very consistent. And again, from that histogram matched set of images, I segmented so that we're only, and masked it so that I'm only measuring the densities from my features of interest. And in this instance as well, I set just one threshold setting for all of them. And here are the results along with the control. With the blue shows the measurements that resulted from the images that I set consistently in Photoshop. The magenta shows the ChromaCal corrected images, and the yellow shows the control. You can see there's quite a bit of variability in the control, where only one threshold was applied to inconsistent images. And again, you can see that the ChromaCal corrected images produced very little variability from image to image, and slightly less variability than those that were set consistently with histogram matching in Photoshop. And again, this is a small sampling, so I'm just showing it for these 4 or 5 images. And remember, and let me not hesitate to say that this is very semi-quantitative, and that doing density measurements from immunoperoxidase stained samples has been shown to not be stoichiometric. In other words, that the uptake is not necessarily linear of the immunoperoxidase. So again, this is for proof of concept, and it's a semi-quantitative result. In conclusion, then, I would say that ChromaCal can be used for creating consistent images, and then for getting morphometry and density intensity measurements. And in this limited sample, we've seen very good results. And so in review, I'd say that, you know, images that are consistent are very difficult to obtain, and it's challenging to set a single threshold with confidence. And in fact, you would probably want to be very subjective and set your thresholds manually, which is, at least for me, it's anxiety producing. It's a subjective method, and it's not, and it's something that I've done in papers, but it certainly isn't something that I feel a great degree of confidence about, although those papers actually have been corroborated. And then to obtain consistent images for analysis, ChromaCal really quickly and objectively calibrates your images to a standard. And I think most importantly, it also indicates whether or not your camera system is producing a linear image. And so if you want to avoid errors in quantification, you know, consistent images are a great way to do that, and definitely a linearity check. Thank you very much for attending. I'll hand this back over to Mark McNulty.
Thank you, Jerry, for, as always, the most informative review of the subject. I do see that we have a number of questions from our audience, and we'll address those in just a moment. But first, just a couple of notes. Our next webinar will cover issues of color and histology in medicine, and we're pleased to have as our guest speaker Dr. Keith Kaplan. Dr. Kaplan is a practicing pathologist as well as respected and vocal authority on digital pathology. He is the principal contributor to the Digital Pathology blog, an online resource to educate and inform the pathology and laboratory community about digital pathology. I also wanted to just briefly mention a current promotion that we're offering at Datacolor through the end of October. You can receive a free ChromaCal calibration slide, and this is an $849 value with your purchase of a ChromaCal system. Details are available by clicking the free slide button on our website at www.chromacal.com. Now for our Q&A session, let me turn the mic over to my colleague, Mark Clymer. Mark?
Thank you, Mark. Okay, so we have several questions that came in. We'd like to address these. Jerry, I'll take this first one. It asks, is a single ChromaCal image used to calibrate images even if an auto white balance procedure is used to adjust white balance for each image in a session? I'd first like to address this by asking that have you ever noticed that if you click white balance in an area of an image and and the camera does white balance, and you click it again, you see a shift. For this reason, we don't recommend the auto white balance settings in your acquisition software. We feel that it's best to white balance at the beginning and maintain that setting throughout your imaging session. However, to answer this question, ChromaCal does include automatic white balancing and brightness adjustment in the images in addition to the color calibration. So it will bring greater consistency into your images, even if you are using automatic white balance in your session. Jerry, here's one for you. So what steps do you take if the camera is not in fact linear?
If the camera is not linear, then you would have to standardize, or you would have to calibrate that against a standard. There are a few standards out there. The one that I'm most familiar with is the Stouffer step wedge. It takes a bit to go through the process, but that would be the way in which you would have to go about it. I think there's a couple of other companies that provide those sorts of calibration routines as well.
Right. So, Jerry, let me follow up on that. Is it, in fact, that the camera is not linear, or is it that the settings that you could be using in the software may actually allow you to acquire a nonlinear image?
Yeah, good question. It's the settings within the software that you'd need to go through and determine which settings to use. Generally, if you set the gamma to 1, you have a linear image, or you produce a linear image, but we found that that isn't always true.
Okay. Here's a question. You talked a lot, Jerry, about linearity. What are the potential pitfalls of using nonlinear images for quantitation?
Well, one pitfall, and this is often what you see in publication, is a comment about whether something is darker or lighter than something else. And if you haven't produced a linear, if you haven't taken, or if the image isn't linear, then you're comparing two things that are no longer apples to apples. You've now changed the gradient so that now something that might have been perceivably much darker than something else is changed. So that would be one issue. The other, again, would be a measurement issue. If you're showing something, if you're measuring from something that does not have a linear gamma, then you can no longer make any comment about whether something is this % brighter or darker than something else.
Okay. Here's another question, Jerry. You showed us examples of color calibration and histogram matching on identical specimen views. Would color calibration with ChromaCal help when thresholding across non-identical specimen images? So I guess take it into a real-world situation where they're not where they're comparing lots of different specimen images.
Right, and I chose just one image field for simplicity, just to be able to see how consistent it is along those lines. But the answer to that is that yes, the whole point behind this is that you would use this on a variety of images over multiple sessions and arrive at consistent images. So yeah, that's and this has been I've done a publication in microscopy today where that was discussed, and but that's the whole idea behind this.
Okay. This is kind of a short question. How do you choose the threshold setting?
Yeah, I mean that's always a challenge. Actually the first paper that I was a co-author on, we would call it a gestalt. We would test ourselves against an image that we used, or an image field that was on a slide. We would test ourselves against that, and then we would cover up the threshold and then try to find the same place and see how accurate we were, and that's really how we went about it. And in this instance, what I did is I just used noise that was introduced when I moved the threshold value, and then backed off until that noise disappeared. That isn't always the best method, but it's one that can work depending on the nature of the image.
Right, and then I guess the best practice would be to use that threshold setting for all of the images in the sample.
If in fact your images are consistent. If they're inconsistent, then you have to change that threshold setting.
Okay.
So.
This is the last question I have in here. So if you have any other questions, please submit them. Earlier in your presentation, you mentioned that monitor color could impact the qualitative assessment of your images. Could monitor color also affect image acquisition?
Yeah, depending on how you acquire your image. I notice that most people acquire their image by evaluating it on the monitor, so they can then decide whether or not to turn the exposure up or down. But if you use a histogram while you're acquiring and you use that as your means for determining exposure, then then the monitor isn't as important. But to my way of thinking, why not know that your monitor is calibrated? To me, that just makes perfect sense. So that, and in response to an earlier question about the auto white balance, actually in the instances that I showed of inconsistency from one image to the other, that's exactly what I did. Is I used an auto white balance for each image, and then just moved the slide over, and I found that there is subtle differences in white balancing, even though I was white balancing against the same area each time. So.
Okay. Thanks, Jerry. There are no other questions in my inbox, so Mark,
Yep.
give it back to you.
Thanks, Mark. For those interested, listed on this slide are a number of resource links on today's topics, as well as contact information for Jerry, Mark Clymer, and myself. So feel free to contact us with any follow-up questions or utilize those resources. So let me conclude by thanking you for attending. Also thanks to our two speakers, Dr. Linden and Jerry Sedgewick. We always appreciate your feedback, so please look for an online survey coming soon regarding this webinar. And as always, a recording of our webinars, and this particular one will be available shortly on the ChromaCal website. So thank you again for attending, and have a great day.